Hi, this is Alex from devstories.eu. AlphaFold, Protein Structure Prediction, Episode 9 of 21. You might assume the biggest hurdle to predicting protein structures is finding a powerful enough GPU, but the real bottleneck is storage. Before you fold a single amino acid, you need to query massive multi-terabyte evolutionary databases. This episode covers deploying AlphaFold 2 locally. Running AlphaFold on your own hardware gives you strict privacy for proprietary sequences and removes the execution time limits found on public servers. DeepMind provides the open-source code directly on GitHub. To use it, you must first replicate the exact genetic reference environment the model uses to find similar sequences. This requires downloading local copies of public databases like BFD, mGenify, UniRef90, and PDB70. Combined, these uncompressed databases consume roughly 3 terabytes of storage. Picture a DevOps engineer provisioning a new high-performance computing node for a research team. The very first requirement is a 3 terabyte drive. You cannot use cheap, slow-spinning disks here. You need fast NVMe storage. Searching through millions of genetic sequences generates aggressive disk read operations. If your storage is slow, the entire pipeline chokes before data ever reaches the neural network. Here is the key insight. The local AlphaFold's pipeline consists of 2 distinct compute phases, and they require completely different hardware profiles. Phase 1 generates the multiple sequence alignment, or MSA. The system searches your terabytes of local databases using external bioinformatics tools. This phase uses exactly 0 GPU cycles. It is entirely bottlenecked by CPU cores, system memory, and disk speed. Phase 2 is the actual structural inference. This is where the neural network takes over to generate the 3D coordinates. And this phase strictly requires a GPU with significant VRAM. If you provision a node with a massive GPU but a weak CPU, your prediction job will stall for hours in the alignment phase. Managing the software dependencies for both of these phases is notoriously difficult. You have to align specific versions of CUDA, JAX, and the sequence alignment tools. To solve this, the official repository relies heavily on Docker. Containerization ensures that this complex execution environment remains isolated and consistent across different operating systems. Setting up the deployment involves 2 primary scripts. First, you run a provided bash script to download the data. You point it at your NVMe drive and let it run. Depending on your network connection, fetching and extracting these files can take a few days. Once the data rests on your fast storage, you are ready for execution. You control the pipeline using a provided Python wrapper script called rundocker.py. You execute this script from the terminal, passing it a few mandatory arguments. You supply the path to the input FASTA file containing your target amino acid sequence. You supply the path to your multi-terabyte database directory. You specify an output directory. Finally, you set a maximum template release date, which restricts the model from using known structural templates published after a specific point in time. The Python wrapper takes these arguments and builds the Docker container. It mounts your local host directories into the container as volumes, allowing the isolated software to read your databases and write output files back to your disk. The container then takes over, orchestrating the CPU-heavy alignment search and the GPU-heavy inference process automatically. Deploying AlphaFold locally is fundamentally an infrastructure challenge, requiring you to balance fast disk speed for the CPU-heavy alignment phase with capable VRAM for the GPU-heavy folding phase. Thanks for spending a few minutes with me. Until next time, take it easy.